hope you all are healthy and safe. And I would like to thank the ACS team for organizing the virtual lecture series. And today I'm going to talk about the In Situ Cycloaddition for the development of small molecule drugs. Particularly, I am going to talk about small molecules for targeting nucleic acid secondary structures. So this is the outline of my talk. That is, first I would like to talk about in situ cycloaddition, which is a target-guided synthetic approach. Then I would like to talk about its application to develop selective ligands for DNA secondary structures like DNA quadruplexes and I motifs. And at the end, I would like to talk about that RNA secondary structure like TAR RNA can also be used as the template to synthesize selective ligands by the in situ cycloaddition approach. So let me begin with the target-guided in situ cycloaddition. So this in situ cycloaddition is a kinetically controlled in situ cycloaddition approach. So this is a target-guided combinatorial approach. In this method, reactive fragments capable of binding the biological target can come into close proximity and then undergo irreversible bond formation to generate triazole products that can strongly bind to the biological target. And thermodynamically controlled dynamic combinatorial chemistry is another approach. And in this approach, reactive fragments capable of simple building blocks assembled through reversible bond formation to generate a dynamic combinatorial library of interconvertible products. And in the presence of a target, the equilibrium of the system shifts towards those products that can strongly bind to the target. Thus, both these approaches, the in situ cycloaddition and the dynamic combinatorial chemistry, both these approaches combine synthesis and screening of ligands, and thus they provide unique platforms for the in situ formation of selective ligands for the biological targets. So this target-guided in situ cycloaddition was first developed by Sharpless and co-workers, in which a biological target selects alkyne and azide fragments and force them to react with each other in a regioselective manner in the absence of any metal-based catalyst. So if we see that the cycloaddition between an alkyne and azide can provide both 1,4 and 1,5 triazole regioisomers. And under thermal conditions, metal-based catalysts have been developed to obtain either the 1,4 or 1,5 triazole regioisomers. But in this approach, the target selects these two fragments and promotes the cycloaddition in the absence of any metal-based catalyst. The enzyme acetylcholine esterase was used as the first target to assemble its own inhibitors from a series of alkyne and azide fragments. However, mostly this method has been used for the protein targets, and it has been less explored for nucleic acid targets. So I would like to talk about the application of in situ cycloaddition strategy for targeting DNA secondary structures like DNA quadruplexes and I motifs. So DNA can adopt many secondary structures. In particular, the guanine-rich DNA sequences can adopt 4 standard secondary structures called G quadruplexes. And as a basic feature, they contain coplanar arrangement of 4 guanines, which are linked together by Hooke's T-type hydrogen bonding. In the presence of metal ions,
Jyoti,
yeah.
Sorry to interrupt you. There is some window on your presentation screen which says, please move this window away from the shared application. So probably move this window because people are not able to see it.
So I don't see anything, but I'm just saying.
Now it's better. Yeah, I think now it's gone. Just
Again it's coming, but
so is this any background screen is open on your computer? Any other window or the page or folder, anything which is open in background?
no, I don't see anything. Yes.
Maybe just full screen it now, because now it's gone completely. So probably if you full screen, it will appear better. Yes, so just full screen. Yeah, awesome. This is great. Sorry to interrupt, but now the view is clear. Awesome. Thanks.
Okay. Yeah, thank you. We were here that gRIS DNA sequences can adopt four standard secondary structures called G quadruplexes, and as a basic feature, they contain coplanar arrangement of four guanines that are linked together by Hookstein type hydrogen bonding. And the story of quadruplex started with a bang. That is, it was Dr. Ivar Bang, a medical doctor by profession, became curious about nucleic acids. And in 1910, he discovered that 5' guanosine monophosphate can self-assemble in water to give gelatinous material. And decades later, it was found out that the gel contains G-quarter-like assemblies, and subsequently, it was found out that gRIS DNA sequences are present throughout the genome. They are found in the telomeres at the end of chromosomes and promoter regions of several oncogenes. And they play key biological functions like telomere length maintenance and regulation of gene expression. So the complementary series DNA sequences can also adopt folded conformations called I motifs. And these I motifs contain intercalated cytosine base pairing due to hemiprotonation of one of the cytosines under acidic conditions. The formation of G quadruplexes and I motifs in the cellular system has been detected by using specific antibodies, like BG4 is specific for G quadruplexes, and the recently developed IMAP can selectively detect I motifs in cellular system. It has been shown that small molecules can bind and stabilize DNA quadruplexes and I motifs and can modulate gene expression in cancer cells. Therefore, small molecules that can bind and stabilize DNA quadruplexes and I motifs can be considered as potential anti-cancer agents. So in our lab, we developed different classes of ligands for targeting DNA quadruplexes. These are peptidomimetics, nucleosides, heteroaromatic ring systems. Many of these compounds show high selectivity for quadruplexes over the duplex DNA. However, the rational design of selective ligands is difficult and challenging because these DNA structures are highly dynamic in nature. And G quadruplexes like c-KIT, c-MYC, HTLO, all of them contain G-quartet as the basic structural feature. Therefore, it is difficult to discriminate between these structures by using small molecules. In contrast to G quadruplexes, only a few ligands are known for targeting I motifs. Targeting I motifs is challenging due to their pH-dependent folding topologies. So to develop selective ligands for a particular DNA quadruplex or I motif structure, we decided to use In Situ Cycloaddition as a tool to generate selective ligands for DNA quadruplexes and I motifs. We synthesized a series of alkyne and azide fragments and then incubated those in the presence of a DNA quadruplex. However, we could not detect any triazole lead as the lead compound. It was difficult to separate the lead compounds from the unreacted starting materials. So thus the identification of ligands was difficult for us. So thus we decided to immobilize the thiolated DNA targets on gold-coated magnetic nanoparticles. We anticipated that the DNA nanotemplates would accelerate the rate of reaction by providing a large surface area and with more number of localized DNA templates. Thus, it could accelerate the rate of reaction. And secondly, the DNA nanotemplates can be easily recovered and recycled for subsequent reaction cycles. These DNA nanotemplates were characterized by using circular dichroism, UV visible spectroscopy, and AIM imaging. So in this approach, we use two different quadruplexes and their complementary eye motifs present in the promoter regions of CMYC and BCL2 oncogenes. Both these genes play key regulatory roles in cancer progression. That is, by targeting potential anti-cancer strategy. So in our approach, we used a mixture of alkyne and azide fragments, which were incubated in the presence of quadruplex nanotemplate. Then after giving sufficient reaction time, we used an external magnet to isolate the DNA nanotemplate along with the newly formed triazole products. The unreacted starting materials remained in the supernatant. The ligand-bound nanotemplate was then heated to release the triazole products, which were characterized by HPLC and mass. Quite significantly, the DNA nanotemplate could be recovered by magnetic separation and could also be reused up to 6 reaction cycles. So we have developed a chemical library, a multi-component chemical library consisting of 4 different alkynes and 17 different azides. The combination of these alkyne and azide fragments could provide 136 possible triazole products, and that are likely to interact with the DNA quadruplexes and eye motifs by using bypass stacking and electrostatic interactions. So each of the alkyne fragment was separately incubated with the azide templates, and the reaction was monitored by HPLC and mass. So in the presence of CMYC quadruplex nanotemplate, only 3 compounds out of 136 possible products were obtained. The lead compound 3AO, which is derived from alkyne 1A and azide 2O, was selected for the CMYC quadruplex, as the other 2 lead compounds were also obtained by the control duplex DNA nanotemplate. The BCL2 quadruplex produced a single lead compound 3AP, which is derived from alkyne 1A and azide 2B. So next, we use the same chemical library to identify selective leads for the eye motifs. So the alkyne and azide fragments were incubated with the eye motif nanotemplates. Then we gave sufficient reaction time, and since the eye motifs are stable under acidic conditions, the reaction was carried out at low pH, and the products were characterized by HPLC and MASH. So 6 compounds were obtained by the CMYC eye motif, and 2 triazole heat compounds were obtained by the BCL2 eye motif. The lead compound 3BE, which was derived from alkyne 1B and azide 2E, was selected for the eye motif, and the lead compound 3BM, which was obtained from alkyne 1B and azide 2M, was selective for the BCL2 eye motif. So these are the hits obtained by the DNA nanotemplates. So by comparing the HPLC traces and the mass analysis, the compounds which are selectively formed by the DNA quadruplexes and eye motifs and are not formed by the control duplex DNA are selected as the selective leads for the quadruplexes and eye motifs. So thus these are the 4 selective leads by the CMYC quadruplex and eye motifs, and which were obtained from a library of compounds that might have generated 136 possible triazole products. And further, these triazole leads were identified as 1,4 triazole leads. So thus this method offers several advantages, like it reduces the time required for the conventional chemical synthesis and their biophysical screening. It also enables easy product isolation and identification, and it also prevents the wastage of expensive commercial DNA, as the DNA nanotemplates can be easily recovered and recycled. Moreover, the resulting triazole ligands show high binding affinities for the DNA quadruplexes and I motifs, and further show promising anti-cancer activities. So now, in the next few slides, I would like to show about the binding affinities of these ligands for quadruplexes and their biological activities. So these 4 compounds were then evaluated for their potential to interact with the DNA quadruplexes and I motifs by using a series of experiments, like we used phosphoresonance energy transfer melting analysis to determine ligand-induced stabilization of folded quadruplex. Isothermal calorimetry was used to determine the binding constants of these ligands for the DNA targets. So this is the summary of the results that I have just summarized these results in one slide. That is, these results are in agreement with the results obtained by the in situ cycloaddition. That is, a triazole lead shows high selectivity for the DNA target that promotes its formation. Like for instance, ligand 3BE, which is obtained by the c-MYC I motif, shows high delta value, that is high stabilization potential for the c-MYC I motif, and not for the other quadruplexes and I motifs used for the biophysical studies. And similarly, it also shows high binding affinities, that is almost nanomolar binding affinities for the c-MYC I motif. So this, to summarize, that ligand 3BE obtained by c-MYC I motif binds selectively to c-MYC I motif, and moreover, it is more potent for the c-MYC I motif in comparison to the other 3 lead compounds. So having assessed the biophysical evaluation of this compound with the DNA quadruplexes and I motifs, we studied the effect of these ligands in the cellular system. So we studied different assays. First, we examined the cytotoxicity of these compounds in cancer cells, and what we observed is that the ligand lead compound for c-MYC quadruplex, BCL2 quadruplex, and c-MYC I motif can inhibit growth of cancer cells without, while showing minimal cytotoxicity for the normal cells. So we use 2 different cell lines, that is c-MYC overexpressing human cervical cancer cell line and BCL2 overexpressing marmoset cell line. We have also used a normal human kidney epithelial cell line. And in contrast to this, the BCL2 I motif lead cannot inhibit the growth of cancer cells as well as the normal cells. So next, further biological studies using FACS analysis revealed that the lead compounds for c-MYC I motif quadruplex and the lead compound for BCL2 quadruplex can induce apoptosis in cancer cells, but the BCL2 I motif lead did not induce apoptosis in cancer cells. Since the G quadruplex and I motifs are promising genetic switches to control gene expression, we studied the effect of these ligands for the regulation of gene expression by using qRT-PCR and Western blot analysis. So these results are here, which shows that the c-MYC I motif lead can reduce the c-MYC expression at both mRNA and protein levels. Similarly, the c-MYC quadruplex lead reduces the c-MYC expression at both mRNA and protein levels. The BCL2 lead upregulates the gene expression in both the cancer cells. In contrast, the BCL2 G quadruplex lead reduces the BCL2 gene expression without affecting the c-MYC expression. So further to examine, so these studies then suggested that these ligands can modulate the expression of c-MYC and BCL2 oncogenes. So to further study whether the effect of these ligands is due to the direct interaction with the promoter regions like quadruplexes and I motifs, we have also studied the dual luciferase promoter assays. So in this assay, we used firefly luciferase plasmids containing c-MYC and BCL2 promoter sequences. And we have also used a firefly luciferase plasmid containing non-GC promoter sequence. And the renilla luciferase was used as the loading control. And these results are also in agreement with the qRT-PCR and Western blot analysis. And we observed that the lead compounds for CMYC and BCL2 quadruplex and CMYC I motif can reduce the firefly luciferase expression, and the BCL2 I motif lead upregulates the firefly luciferase expression. So this is the summary of these results of qRT-PCR Western blot and promoter luciferase assays, which shows that the lead compound for CMYC quadruplex and CMYC I motif can reduce the CMYC expression without affecting the BCL2 gene expression, and the BCL2 quadruplex lead did not alter the CMYC expression but reduced the BCL2 gene expression. The CMYC quadruplex and I motifs and BCL2 quadruplex would be a potential strategy for anti-cancer therapeutics. In contrast, the BCL2 I motif lead upregulates the BCL2 gene expression. Therefore, targeting BCL2 I motif could be useful for neurodegenerative disorders that involve cellular death. So, thus to summarize, the in situ cycloaddition is very useful to develop target selective ligands and that's so promising biological activities. So next, I would like to show you the dynamic combinatorial chemistry, as I have mentioned previously, that this is also an approach to generate selective ligands for quadruplexes, and this is also a combinatorial approach. So in this approach, we used aldehyde and amine building blocks that reversibly react to generate interconvertible imines. In the presence of quadruplex, the system evolved to generate quadruplex selective imine binders, which were then reduced by to give the corresponding amines. The amine-bound DNA nanotemplate was separated by magnetic decantation and again heated to release the quadruplex selective amine ligands. Using SPLC and mass analysis. We also carried out a control experiment using duplex DNA nanotemplate. So in this approach, we used a carbazole aldehyde and 10 different amines that reversibly react to generate interconvertible amines. In the presence of quadruplex nanotemplate, the system evolves to generate the quadruplex selective imine binders. That is, the G quadruplex nanotemplate selects its best imine binding ligands, and then these imines were reduced by sodium cyanoborohydride to give the stable amines. So this is the HPLC analysis, that in the absence of any DNA template, all possible amine ligands were obtained, and in the presence of control duplex DNA, 5 different amines were identified, and in the presence of quadruplex nanotemplate, only this carbazole amine was obtained as the lead compound. So biophysical studies using plate melting and fluorescence binding titrations revealed that this carbazole lead compound shows high selectivity for the quadruplex over the control duplex DNA. Further biological studies revealed that this ligand is toxic to the cancer cells, non-toxic to the normal cells, and it can also reduce the CMYC expression in the cancer cells by directly stabilizing the CMYC quadruplex structure. And further confocal microscopy reveals that this ligand is cell membrane permeable and mostly localizes in the cellular nucleus, and it can also induce DNA damage response in cancer cells. So this is the first example of dynamic combinatorial chemistry using a DNA nanotemplate that selects its best binding ligands from a dynamic library. So I have shown you the two different approaches, the in situ cycloaddition, that it can be used to generate selective ligands for quadruplexes and I motifs. And in this approach, the target promotes the azide and alkyne cycloaddition in the absence of any metal-based catalyst. And in dynamic combinatorial chemistry, we used aldehyde and amine building blocks that reversibly react to generate imine-based dynamic combinatorial library. So in the last part, I am going to present the in situ cycloaddition using TAR RNA as the template. The TAR RNA, that is the trans-activation response RNA, is found in the LTR sequence of the human immunodeficiency virus type 1, that is HIV-1 genome. And the TAR RNA can fold to form a stable hairpin structure with a hexanucleotide loop and 3 base UCU bulge region. The bulge region of the TAR RNA is the binding site for the TAR peptide, and the interaction between the TAR and TAR is essential for the viral replication. TAR RNA, therefore, has been targeted by small molecules to inhibit TAR and TAR interactions. As a result, the viral replication is inhibited. Therefore, small molecules that can bind and stabilize the TAR RNA are considered as antiviral agents. So these are some of the reported ligands like antibiotics, aminoglycosides like neomycin B, and different heteroaromatic ring systems are known as the previously reported as TAR RNA binding ligands, and they are also potent antiviral agents. So given the dynamic folding topology of RNA structures, the in situ cycloaddition could be a potential strategy to develop RNA selective ligands. So therefore, we use TAR RNA as the target template, and we use TAR DNA and TAR RNA without this UCU bulge region as the control template. Then we developed a multi-component library consisting of 3 different alkynes and different azares. Particularly, the alkyne fragments were derived from thiazole ring system, which is a 5-membered heterocyclic ring system, and which shows potent biological activities and present in numerous natural products and biological active compounds. So this is the schematic representation of our templated cycloaddition. That is, the alkyne and azide fragments were incubated with biotin tagged TAR RNA. We used here biotin tagged TAR RNA so that it can be easily separated by streptavidin beads. And then we give sufficient reaction time for the templated cycloaddition, then use streptavidin magnetic beads to isolate the TAR RNA along with the newly formed triazole products. The unreacted starting materials remained in the supernatant. And then by magnetic separation and heating, we could separate the triazole products, which were then characterized by HPLC and mass. So this is the HPLC and mass analysis that the TAR RNA produced 2 different compounds. One is ligand 3BA, which contains two thiazole rings and 3 thiazole rings. This is as the minor compound. This is the major compound, is the 3BA. And with TAR DNA, that 3CA was exclusively obtained, and the TAR RNA, in the presence of TAR RNA, the ligand 3AA was formed. So this is the summary of the in situ cycloaddition results, that compound 3AA that contains 1 thiazole ring was obtained exclusively by the TAR RNA without the bulge, and these 2 compounds are obtained by TAR RNA, and the compound which contains 2 thiazole rings and the flexible CH2 ring is considered as the selective lead for TAR RNA, as the other compound was formed by the TAR DNA. And these studies also suggest, since these 2 compounds are not formed by the TAR RNA without the bulge region, that these 2 ligands possibly binds in the bulge region of the TAR RNA. So then the interaction of these 3 ligands with the 3 nucleotide sequences was studied by using a series of biophysical experiments, and I have summarized only the lead compound, that is isothermal calorimetry and fluorescence data for the lead compound 3BA. And this ligand binds to the TAR RNA with a sub-micromolar binding affinity and 1:1 stoichiometry, and it was also identified as the major compound in the in situ cycloaddition. And the biophysical studies also suggested that the ligand 3BA is more potent for the TAR RNA. And we further assessed the ability of these ligands to displace TAR peptide from TAR RNA by using a thread-based assay. So in this method, we used FAM-labeled TAR RNA, that is donor-labeled TAR RNA, and an acceptor-labeled TAR peptide. So, and the fluorescence of this donor fluorophore was monitored by fluorescence spectroscopy, and the study reveals that the ligands could displace the TAR peptide from the TAR RNA. And this further suggests that these ligands bind in the bulge region of the TAR RNA, which was further supported by the molecular modeling studies. So thus, a new class of peptidomimetic ligands has been developed for TAR RNA by in situ cycloaddition. So this is my last slide, that to summarize that the in situ cycloaddition can be used to develop selective ligands for DNA and RNA secondary structures. And as already mentioned, that this method combines both synthesis and screening, and therefore it can bridge the gap between synthesis and screening, and it also enables rapid synthesis and high throughput screening. And moreover, it can be used for targets whose structural details are not known yet, and it can also be used where the rational ligand design is difficult. However, most of the studies are proof of concept studies. New and challenging targets needs to be explored, and new bioorthogonal reactions should be developed for generating more specific and complex lead compounds. And finally, I would like to thank my research scholars for their hard work, and cooperation and funding agencies for financial support. And I would like to thank you all for your kind attention. Thank you.